Adenylyl cyclase is part of the pain pathway. Although advancements in the understanding of the molecular mechanisms of adenylyl cyclase have been made, the same cannot be said about the applications of these mechanisms in pain management. Wang and colleagues began research on how adenylyl cyclase can be targeted as a therapeutic alternative to opioids, which can be addictive and increasing tolerance is gained over time, making these drugs harmful.
Mammalian membrane-bound adenylyl cyclases have a twofold symmetry with a pair of class III cyclase homology domains. Class III cyclases are generally integral membrane proteins responsible for translating external signals into intracellular responses. They are regulated by G proteins, which can either be inhibited by G-alpha-I proteins or activated by either stimulatory G proteins, G-alpha-S, or forskolin, with the exception of adenylyl cyclase 9. The adenylyl cyclases function as enzymes which catalyze the conversion of ATP to cyclic AMP. Cyclic AMP serves as a ubiquitous secondary messenger and is known to regulate a variety of cellular processes, including transportation, enzyme regulation, and sensory transduction. Humans have 9 different isoforms of membrane-bound adenylyl cyclases, named AC1 through 9, which differ in regulatory mechanisms and expression.
Some groups of adenylyl cyclase are activated by calcium ions and calmodulin, while others are inactivated by calcium. AC1 and AC8 are regulated by calcium ions and calmodulin and are expressed in the hippocampus. The hippocampus is a region of the brain associated with memory. A lack of both AC1 and AC8 can lead to memory loss. However, mice without AC1 gene exhibited less pain sensitivity and opioid dependence. Because of this discovery, AC1 could potentially be a target for pain medication since there is no memory loss as long as AC8 remains uninhibited. The Wang researchers found 2 compounds with the potential to be used as analgesics, as they both inhibit AC1. The first compound, STO34307, is selective for AC1 inhibition, while the second, NB001, was less selective and its mechanism of inhibition is indirect. The researchers investigated both the effectiveness and specificity of STO34307 for inhibition of AC1. They found that STO34307 was able to inhibit the accumulation of cyclic AMP in HEC-AC1 cells, and also found that STO34307 did not have any inhibitory effect on other ACs, even at high doses.
In summary, ST03437 is a chromoin-derived small molecule that is highly selective for AC1 inhibition. The inhibition is mechanism-specific, and most likely does not inhibit G protein subunits. In the central nervous system, the N-methyl deaspartate receptor is hypothesized to provide influx of calcium ions, leading to activation of AC1, long-term potentiation, and ultimately pain response. By specifically inhibiting AC1 directly, this pain response can be reduced.
In conclusion, STO34307 has great potential to function as an analgesic to treat both inflammatory and chronic pain. It also has potential as a complement for opioid drugs, whereby a lowered dose of opioids can be combined with STO34307 for a greater pain-relieving effect.